Remember that one of the qualities of genetic material such as DNA is that it's able to express information. And DNA does that by producing proteins. And proteins, as you remember, are things like enzymes. They provide structure. They kind of take care of everything inside individual cells and inside organisms. So there are two parts to protein synthesis: transcription and translation. We're going to focus first on transcription, when the DNA message is transcribed or coded into messenger RNA. So we're looking at this part right here. The next video will focus on translation. Overall, transcription is a fairly simple process compared to translation, which we'll get to next. What you have to keep in mind is that the DNA that's making up your chromosome, so here's our DNA in red and blue, is made up of lots of different genes or different segments within the DNA that code for different proteins. So during transcription, the first thing that happens, so first is DNA helicase, right, which we looked at with DNA replication, right? DNA helicase comes along and unzips the double helix of the DNA by breaking the hydrogen bonds between the nitrogen bases. And it doesn't unzip the entire DNA molecule, it just does that region, that gene that is going to code for a protein. The second thing that happens is that this enzyme right here, which is called RNA polymerase, we talked about DNA polymerase before, now we're looking at RNA polymerase, it manages to read the DNA that has been exposed. So in this case, the blue strand of the DNA is acting as what we call the sense strand, or sometimes it's also called the template strand. Okay, so sense or template strand, and it's matching up base pairs. We'll talk about how that happens in just a moment. And then finally, the third step is that your RNA molecule, your messenger RNA molecule, which has been transcribed, leaves the nucleus through the nuclear pores, and then it's going to go into the cytoplasm and find ribosome. That's translation. So it gets unzipped, right? Unzipped, zoop, build a new RNA molecule, messenger RNA, by RNA polymerase, and then it leaves the nucleus because the DNA cannot leave the nucleus. Okay? So that's what's really important. We need to get that message from the DNA to the ribosome where proteins are made, and in order to do that, messenger RNA comes into play. Remember, the RNA polymerase matches up the new ribonucleotides, the building blocks of the RNA, to the DNA code. So if we do our DNA in red, right, so we have 4 possible bases, A, T, C, and G. The RNA that matches up, okay, G's and C's are still exactly the same, so C and G are complementary bases. If there is a T, thymine, in the DNA, the complementary base pair of the RNA is adenine. And if there's an A, adenine, in the DNA, this is the difference. Remember that in RNA, there is no thymine as a nitrogen base, but instead we have uracil. So the U is for uracil. So notice, like right in here, okay, this first one. The DNA was adenine, so the RNA that's made is uracil. So if you ever see a long code of nitrogen bases, if you see U's instead of T's, that means you're looking at RNA. So again, our overview: during transcription, the DNA message is encoded into mRNA, so we can see that happening here. Right, here's our mRNA molecule being made. And once that happens, that RNA is going to leave the nucleus. Here's our nucleus of the cell, right? Leave the nucleus and go to the cytoplasm and find a ribosome. And we'll look at what happens in translation next.